Welcome back to our Systems Biology podcast. In this podcast, I meet people from academia or industry who are working to understand biology using maths and computing. They come from different academic background, professional backgrounds, but all of them are trying to understand using life and life science using tools of math and computing. Today, my guest is Dr. Hamim Zafar. He's a faculty at IIT Kanpur. Hamim is a faculty in two departments together in Biosciences and Bioengineering. At the same time, he's a faculty in the Computer Science department. Welcome, Hamim, to our Systems Biology podcast.
Thank you, Biplav, very much. Thanks very much for inviting me to your podcast. Very excited to be here.
So before we go into discussion about one of the very exciting area of Hamim's work, let me tell the audience briefly to create the excitement. See, till now there are lots of questions in life sciences which are unresolved, and some of them matters life and death. And one such problem is cancer biology. Cancer still is the second largest cause of death globally. So there are lots of open-ended questions in cancer, and people are trying to understand cancer biology from different perspectives. Hamim is one of them. He is trying to understand biology of cancer cells using the computing tool algorithms that he develops and his colleagues develop. So let me give the audience a brief about the Hamim's academic journey. Hamim, you did your BE in electronics from Jadavpur University, Kolkata.
Yes.
And then you moved to Rice University, US, just like many other bright students of our country, and did a PhD in computer science there. And then you returned back to India. In between, you went to Carnegie Mellon as a postdoc also. And now you work as a faculty both in the computer science as well as Bioscience and Bioengineering department. And you work on the cutting edge, using cutting edge tools to understand the evolution of cancer cells. So my first question would be, I think many of our audience has the same question in your mind. Are you an accidental biologist, or you still consider yourself as an engineer trying to find solutions about questions in life?
Yeah, that's actually very relevant question. So I would say I'm neither an accidental biologist nor solely an engineer. At this point, I think I'll call myself as a computational biologist. So I'm basically an interdisciplinary researcher, and I'm trying to solve questions in biology with the help of computation. So as you pointed out towards my journey, right? So I did my undergraduate studies, which was in electronics and telecommunication from Jadavpur University. And even at that point, again, of course, as an engineer, I was learning different concepts in, let's say, signal processing, algorithms, and so on. In fact, I did a little bit of research work as well. So that time I was working on stochastic optimization. And that actually eventually got me interested to pursue a PhD in computer science. So when I moved to Rice for my PhD, my major goal was to work on machine learning. So I think machine learning was a very hot topic around that time. So that was definitely one of the aspirations. So what I found out after talking to different faculty members at Rice University, I found it very interesting, the research group of Professor Luai Nakle, who eventually became my PhD supervisor. So I saw that they are working on, again, applications of machine learning, but they are working with biological data sets. So not like toy data sets, image data sets, or text data sets that are more common in the field of machine learning, but these are actually data sets generated from biological experiments, right? So that got me intrigued, and eventually I started working with him. And that, you can say, was sort of my journey into computational biology. I started working in computational biology, and it was fascinating to see these two very different fields meet, right? So computer science, usage of computer science mathematical models, again, to solve biological questions. And during that journey itself, again, I started working in collaboration with some scientists from MD Anderson Cancer Center, and that was sort of my journey into genomics per se. So we started collaborating with Professor Nicholas Nevin's lab and Professor Ken Chen's lab. In fact, Professor Ken Chen became my co-supervisor as well for my PhD. And that time, single cell sequencing was at the, I would say, at its infancy. And Professor Nicholas Nevin's lab, they actually developed a lot of assays. They still develop a lot of assays in single cell DNA sequencing. And I found that problem very interesting, that they were actually trying to identify somatic mutations from these newer type of data sets. So before single sequencing, everything was sort of like bulk sequencing data. Again, we can come to that later, but at that point, I found the problem very interesting, particularly because of its application in cancer. Right. So we know cancer is a very deadly disease, and I thought it would give me a very first-hand chance to work with patient data sets generated directly from the patients, and maybe I can develop algorithms to solve some questions which can actually matter in life. So that was very fascinating, and that's how I started my journey. So with time, of course, I have diversified. I don't work only on single cell DNA sequencing, but also on other types of omics datasets as well. But yes, cancer remains one of the main interest in the lab. And with all these things in mind, I think, again, by training, I am a computational person. I am a computer scientist or an engineer, you can call. But then again, the research questions that I am looking at, they are deeply rooted in biology. So yes, I would call myself as a computational biologist seeking to find answer to different questions in biology, but my tool sets are computational.
Oh, nicely put. So, as you said that you worked along and even still work on to some extent on this single cell DNA sequencing, and I
Yes.
also reading your papers. Also, a bulk of that is actually single cell data. I'll come to that because I'll go into details of that. But just a quick question here. So, when you were in Jadavpur studying electronics, do you ever had any clue that in biology people are using these AI/ML models, statistical inference models? Do you ever had a clue?
At that point, no, definitely not the case. At when I was in Jadavpur University, I never knew that in biology people actually use this kind of mathematical models, because I was more familiar with usage of this kind of models in signal processing, or let's say antenna design fields like those, because those were what I was exposed to. It's only after moving to Rice University I got exposed to this field of computational biology.
So your life has taken you to different exposure, and you
Yes.
either taken to a new journey.
Yes.
That's what we should be. All should be open to new journeys actually.
Yes.
Otherwise, we are stuck in one place. So coming back to your area of work, we will discuss at length that how actually that is you are using those techniques and tools to understand cancer biology. So you are saying that these are single cell genomic data. I mean, essentially, these are single cell data. What do you
Yeah.
mean by single cell data? I mean, and why it is so important? What is the difference between that and traditional biology? Is there any difference?
Yes, that's definitely a very pertinent question. So, single cell biology, again, it's a new and evolving field in the field of biology. So you can think of that now we are able to generate data from individual cells. So before single cell, if we talk about genomics in general, people actually generated data in a bulk. So you have a tissue consisting of millions of cells, and you basically do one measurement from the whole tissue, right? So that is what is known as bulk omics data. Again, if we are studying DNA, then it's genomics. If we are studying RNA, whole RNA of the tissue, then it is transcriptomics, and so on, right? But now with this bulk data, the problem is that what you are getting is an average signal, right? Most of the biological systems are very, very heterogeneous. So we know that any particular tissue, let's say liver, or let's say heart or muscle or any other kind of tissue, we know that there are different types of cells, and these different types of cells, they have different types of functions. They might vary in terms of morphology, they might have some other kind of properties as well. But if we are doing a single measurement, which is sort of taken as an average of all the different cells in the tissue, then that is not going to give us a very good insight about this heterogeneity, right? Of course, it is overall we are getting some signal and we have some understanding, which is also good in a lot of cases. But again, if we really want to go to the nitty gritty details of how cells are actually working together in a particular tissue context, then we need to actually look at individual cells. And this is very, very important. So think about, the analogous thing would be to an orchestra. Suppose you have an orchestra where a lot of different instruments are being played, and but you are not able to distinguish the instruments. What you are seeing is an average symphony, right? But now with this kind of single cell omics data or single cell sequencing, now you can actually pick up every individual signal separately. So you know which one is coming from drums, which one is coming from guitar, which one is coming from piano, and so on, right? So again, these musical instruments are just analogous to different types of cells that might be present in that heterogeneous system. So again, I think in the past 10 years or so, there has been tremendous development in lot of biotechnological assays, where now we can actually get very high throughput data sets. So imagine that you are looking at the complete DNA sequence of individual tumor cells. We know cancer is actually very heterogeneous. So it's not the case that cancer consists of one type of cancer cells, right? Even inside the cancer tissue, there are multiple types of cells. One group of cells might be having one type of property, another group of cells may have another type of property, and sometimes they vary in terms of their molecular profiles as well. So maybe the mutations are different, maybe the genes that are actually activated, or the different gene programs that are activated in these cancer cells, they might be different. So to understand or to study this kind of heterogeneity, we need a single cell level data. So that's what this single cell sequencing technology actually can give us. And it has been tremendous. There has been tremendous growth. So if we look back to, let's say, somewhat like 2013, 2014, 2015, around that time, technologies could actually get data from, let's say, 100 cells. But now we are actually looking at data sets consisting of millions of cells. So this is actually huge amount of data. And with this data, we can now truly profile the heterogeneity. And at the same time, we are profiling for, let's say, thousands of genes, not just a single gene or 200 genes or 100 genes, but rather the whole transcriptome can be profiled. So what we are getting is gene expression profile of these individual cells. So imagine that in the cancer tissue, along with cancer cells, we also have stromal cells, we have infiltration of immune cells, and all of these cells together, they are actually part of a very interesting ecosystem, right? Which we call as tumor microenvironment. And this tumor microenvironment has a lot of role to play in different phenotypes. For example, in some patient, it might actually promote cancer. The kind of cells that are present in the tumor microenvironment, they might actually promote the progression of the tumor, maybe promote even metastasis, which is when cancer cells migrate from a primary location to a secondary location. But similarly, there might be a different patient where the role of the cells are completely different, like they are producing different types of molecules, and maybe immune cells are active enough to kill the cancer cells. So cancer is not able to spread that much. So this type of heterogeneity can only be studied through single cell sequencing.
Okay, so if I gather what you are trying to say in a layman term is that, see, if I understand cancer thing a little bit, so
Mhm.
if somebody has a solid tumor,
Yeah.
the pathologists take a biopsy sample,
Yes.
right? So they
Yes.
get a tissue sample, isn't it? So
Yes.
that tissue you are saying is made up of millions of cells. It is not
Yes.
just one cell. So
Yes.
traditionally, you are saying that when they will take the biopsy tissue sample, they will try to do an assay,
Hmm.
some sort of assays by which they will know the overall property of all the cell together.
Together. Yes.
And what you are arguing is that even if I take a small tissue, that does is not made up of identical cell. There
Hmm.
are different types of cells,
Yes.
and there are also, apart from cancer cell, as you said just now, there are some normal cells also,
Yes.
right? So if
Yes.
I take the traditional assay where the average I am getting, I
Hmm.
am missing the whole picture.
Yes.
So you want to see now individual cells behavior separately. That's
Yes.
what single cell thing is coming, right?
Yes, exactly. And I
Okay,
will give you
okay.
one particular interesting idea also, like, or maybe why we need to study this kind of heterogeneity, right? So let's take the
What do you
example.
mean by heterogeneity actually in this case? Can you tell us actually?
So heterogeneity is basically that within the same tissue, you are seeing amalgamation of different types of cells, right?
Okay.
That it is not a single type of or identical cell population, but the cell population is consisting of diverse set of cells. This is what I'm calling as heterogeneity. So why
Okay.
is it important? Suppose in case of cancer, we know that relapse is very common, right? So what
Okay.
actually happens? Imagine you have two types of cancer cells in your cancer tissue. You administer a particular chemotherapy, and that chemotherapy is only able to kill one type of cancer cell. The
Right.
other
Right.
type of cancer cell is actually sitting quietly, and
Mm-hmm.
initially it might seem that, oh, patient is responding very well to the treatment, cancer is actually diminishing, and
Mm-hmm.
maybe 6 months down the line your cancer almost vanishes, you don't see
Mm-hmm.
it. But again, 1 year or 2 year later, you see
Mm-hmm.
cancer again relapsing. Why? Because there was a quiescent small population of cells which were not killed by the initial therapy, right?
Ah.
So that's where it gets very important.
Okay, okay. So now I get it. So you are saying in the tumor, actually there are subpopulations. So the drug may
Yes,
have killed majority of the population, but some notorious cell may have survived, and then
yes,
after 1 year they grow and create the tumor again, and the patient is back to hospital. That's what you
yes,
are trying, right?
exactly,
So
exactly.
if you can have, if you have done individual cells assay at the early stage, possibly you could
Yes,
have got those notorious thing identified early.
exactly. Yes.
Okay, okay. So
So
I was just reading. I have not gone details about all these your papers. So what I gather even now, as you are saying, so the crux of the baseline of your work relies on this idea that when you take a tumor, it is not homogeneous thing. It is a subpopulations are there, and you use genomics, mean DNA sequence analysis, RNA sequence analysis data from individual cells to reconstruct how, as I was reading in your paper, is the evolutionary pathway, something
yes,
like that, right? So can you just explain that to our audience? I mean, what you are doing, what are you inferring, what are you trying to learn?
absolutely, absolutely. So let's take the example of cancer itself, right? Though we work on other kind of biological systems as well, but majority of our focuses have been on cancer. So as I said, cancer consists of distinct tumor subpopulations. So that means different types of tumor cells. And when I am saying they are distinct, I am not just talking about their morphology or anything, but I am talking about their molecular properties. That means let us characterize them somehow. How can we characterize the cancer cells? We know that cancer is caused by somatic mutations. So these are
What is
basically
somatic
changes.
mutation in that?
Yeah, so somatic mutations are basically changes in your DNA sequence,
Okay,
right?
okay.
So these
Mhm.
are known as mutations, and they can be of varying length. It can involve just a single position of DNA, or it can also involve multiple positions. So,
Mhm.
anyways, now we are saying that these cancer cells are there, and because cancer is caused by somatic mutations, that means somatic mutations can be different. The profile, mutation profile, can vary from one cell to another, right? So, one of the first tasks or one of the first thing that we do in this scenario is that we want to characterize the tumor populations with respect to their somatic mutation profile. That means, suppose I have got a data set of thousands of cells. Those thousands of cells, now I will first try to identify the somatic mutations in each of them. And then, with respect to this somatic mutation profile, I will try to group the cells. Maybe I will end up with two, three groups per individual patient, right? So, this is one level of characterization. Then comes looking at the evolutionary history of the tumor. Because what is happening? We are getting biopsy sample, which is basically one time point during the tumor evolutionary process. So tumor is actually a very dynamic system, right? Over time, it basically again tumor over time it develops first develops from the normal cell, but it is under continuous selection pressure. So that's why a lot of changes are happening dynamically. So you can think of it as an system, a particular system under evolution, undergoing Darwinian evolution. And what we are doing, we are just taking a snapshot of this dynamic process, because at the biopsy, when the biopsy is done, we are taking a sample and we are trying to look at that particular sample. So a lot of things have already happened in the past, things as in events, as in different mutational events, which actually led to the current state of the tumor. right? So one of the very important tasks with this kind of data is that to reconstruct the evolutionary history of the tumor cells. That means how and when actually tumor started spreading. How, what was the first initial mutation that probably would have moved or shifted the cell from normal to tumor, and then how did it acquire subsequent mutations, right? So in the parlance of again this tumor phylogeny, so what we are trying to look at, we are trying to reconstruct the tree structure which might actually give us the idea that how tumor evolved over time. So we are calling it as a tree structure because again there are different branches that could have happened during the past, right? So one subpopulation was there, and because it acquired new mutations, it got divided into, let's say, two different branches. So this kind of branching actually happens during the evolutionary history of the tumor, and now we want to reconstruct everything from the data. So whatever we are going to reconstruct, this is known as tumor phylogenetic tree. Okay, and in this tree structure we have, suppose we have this individual tumor cells at the leaf level, and then lot of different internal nodes basically refers to ancestral population, and ultimately we have the root. The root can be defined in terms of, let's say, difference with a normal cell. So whenever single cell data is generated, you can also imagine that some cells will also be normal, they are not tumorous, right? So those normal cells will be on one side of the root, and all the other tumor cells will be on the another side of the root. So in this manner, we are actually able to reconstruct what are the very early mutations and what are the late mutations. And it is very important that we understand both, because what can happen, some of the early mutations probably initiated cancer, right? So if we know and we can find a pattern across different patients, then probably some sort of therapy can be designed to target those mutations, which we are actually calling as precision therapy or precision medicine. And similarly, at the late stage, when we are actually looking at mutations that happen late, and probably they are responsible for cancer progression, right? So it might happen that at certain stage, a new group of cells appeared with a distinct type of mutation, let's say in a particular pathway, and that actually overtook the complete tumor tissue, right? So that became a very dominant population. So we know that it is an aggressive dominant population, and we need to handle it somehow. So this kind of tumor reconstruction of this tumor evolutionary history actually can help us with this kind of questions.
So if I understand correctly, so it is almost equivalent to something like that. That, as we say, in a school we teach or we discuss that, okay, human have originated from these organism.
Yes.
So the chimpanzees and human has a common ancestor from which we branched out. So you are saying the
Yes.
similar thing, that when I take a tumor sample, a doctor has taken a tumor and seen that tumor, those tumor cell has actually originated somewhere in the patient body through
Hmm.
that type of evolutionary thing. So there are common ancestor and all these things, right?
Exactly.
So it is almost
Exactly.
similar to that idea, isn't it? So
Yes.
so
Yes.
I always had two questions. In fact, I haven't got opportunity to discuss at length with some evolutionary biologists, but I always had the idea to see you are looking at one single time point,
Yes.
right? From that, I can actually calculate what has happened in past. I mean, that is
Uh,
possible.
yes, that is possible.
Okay.
So, and the field of looking at this evolutionary history, that is basically the field of phylogeny. Now, as you
Okay.
rightly pointed out, that it is very similar to like different species evolving together, right? Like human, chimp, gorilla, and we share a common ancestor. So, how do we know that we have a common ancestor? We look at the DNA of this individual species, and then we see there are very much similarity across these organisms, across these different species. The DNA is very much identical, right? There are some differences, and these kind of differences are again like when we are studying species, these differences are coming due to a lot of different events in genetics, like again speciation, let's say gene duplication, loss, or even there are other kind of events where actually you can get some sort of divergence in your gene, functional divergence, and so on, right? So ultimately, the study of this, how different species actually evolved together, that is phylogeny. And because now we are trying to apply the same concept in the context of cancer cells or tumor cells, that's why we call it a tumor phylogeny. So here we do not have species, but rather instead of species, you can imagine that we can think of tumor subpopulations as sort of the unit of this phylogenetic tree, and the relationship between them will be dictated by this kind of different mutations that have happened over different time scale. So yes, we still have a snapshot data at a single time point, but because the mutational profiles vary from one cell to another, these mutational profiles are our signal for reconstructing this tumor evolutionary history.
So if I get it, essentially the role of experimental biology is ended there to give you that data.
Yes.
That experiment has not told you the tree, it has not told you the evolutionary path. Your
Hmm.
mathematics is telling what is the path,
Yes.
right?
What is
So
the
without
most
that
likely?
mathematical or statistical model, we do not
Yes.
have that path history at all.
Yes.
We only have one point data,
Yes.
isn't it? Yeah.
Yes.
So
Exactly.
so
Absolutely.
that must be a quite a powerful mathematical tool that we can reconstruct, right? So
Yeah.
can we use this to even predict what will happen in future?
In some cases, yes, but particularly with this evolutionary trees, we cannot really predict what kind of new mutation might appear, because again, the model is trying to reconstruct the past, but not really trying to predict the future. So
Okay.
for predicting the future, you may have to look at other set of mathematical models,
Okay.
right? So
But that type of argument, the formalism exists, right?
In some problems, yes. For
Okay.
example, what we can say is that in the field of single cell RNA sequencing. So now let us shift the gear a little bit towards transcriptome data rather than DNA data, right? So because in transcriptome data, at least this is more prominent, this kind of models. So now you imagine that you are looking at a cell population and we are trying to look at their gene expression data. What is gene expression data? It's basically telling us how much a particular gene is active in a particular cell. So we are just looking at their transcript counts in individual cells. So you have this data. Now what you have done, suppose you have tried to fit a mathematical model, which in this case is a neural network, right? So and there is a specific type of neural network also. Let's call them as variational autoencoder. So what this model has given us how different cell populations cluster according to their gene expression profile. Okay. So now each cell is represented by a lower dimensional vector. Okay, because we started with a very high dimensional data. Let's say for each cell we have a measurement of 20,000 genes, but now through this mathematical modeling I am able to represent each cell as a 20 dimensional representation. It's a numerical vector representing the biological identity of the cell. Now, as you rightly pointed out, that what can happen given some sort of perturbation or something, right? Now you can actually model the perturbation in this latent space. So if
Okay.
you think that a perturbation is given, now a perturbation is given, and that might actually change uh, some particular pathways, right? Response of some particular pathways. So what you would do some linear arithmetic or some other kind of arithmetic in the latent space representation. So what happened? You had one cell, and then through this mathematical modeling, you are now getting the state of the perturbed cell, right? Now you take that state and you pass it through another neural network. Let's call it a decoder, and it is going to reconstruct back the 20,000 length 20,000 vector where each entry is corresponding to individual genes. So now what you are able to do with this kind of model is that you are able to figure out the effect of certain perturbation. Again, it's predictive, so it will not probably work all the time correctly, 100% correct, but it can give you a ballpark of the idea that what possible pathways could be dysregulated because of this perturbation.
so if I catch the keywords from what you said, it seems like this is an area, I do not know, you correct me, is a area of what we traditionally call statistical inference or statistical learning, this type of work, isn't
Uh,
it?
Statistical inference, but then again I would place it in the league of, let's say, deep learning, because again we are
Okay.
using neural networks. So it's
Oh, in
basically
these
a
cases.
marriage. This is basically a marriage between deep learning and your probabilistic inference. What is
Okay.
happening is that ultimately we want to understand, or we want to learn the distribution of the data points. So we are assuming some sort of probability distribution. We are trying to estimate their parameters and everything, but this estimation and everything is happening with the help of a neural network.
Okay. So as you said, it is very interesting. You're saying it is not just single cell data. You just said few minutes back that if you
Hmm.
take a biopsy sample that may have millions of cells,
Yes.
now you are saying from that cell, you are collecting data of 10,000, 30,000 genes.
Yes.
So that means 1 million into 30,000. So it's a
Absolutely.
huge data, huge
Huge
data.
amount of
Your
data. Yes.
Excel sheet will never be able to handle it, right?
Yes.
So yeah, many of my colleagues were telling me, see, actually the biology has become high throughput biology, higher dimensional
Absolutely.
data.
Yes.
You are
Yes.
generating data like this. When I was doing PhD, we used to take 3 days to do the one single gene sequencing, and
Mhm.
then you read it manually. It take 1 week to get the sequence, and you may land up at a wrong sequence actually because you have messed up the experiment. But right
Mhm.
now, I think they do it in a day, the whole genome and all these things, isn't it?
Yes.
So it is high
Exactly.
throughput, high dimensional, and
Mhm.
as I understand that, to understand and make a meaning out of it, traditional analysis does not work, and that's where people like you come into play an important role to create algorithms to understand these data sets and make a meaning out of it, make a prediction about it, right? So
Yes.
one thing still actually many people are, have a concern is that, is it that role of mathematics is primarily remaining in analyzing and visualizing, curating this data, or rather biology is becoming fundamentally more math driven? So these are two
Uh,
different aspects, isn't it? Obviously, if you have large data, you require better visualization. You
yeah.
require better, computing facility to store and handle that. Right? My 4 GB RAM will not work anymore. We need, right? So is it only that issue, or actually computational biology or mathematical biology is actually leading biology to more quantitative and maths-driven science? So where do you think we are moving?
So I think both the aspects that you just described, I think to some extent both are sort of true. So if you think about the first thing, definitely biology has become much more quantitative these days. Just because we now have the measure capability to measure so many genes in parallel, or so many samples in parallel. So for that, essentially, like as you mentioned, that traditional biological experiments focused on a single gene, what would happen if we perturb that gene, and so on, right? Now, but now you are actually looking at the systems level changes that can be again. So you do a perturbation, but you are not measuring a single gene, but rather you are measuring all 20,000 genes, right? So certainly, because the data is high throughput, we need quantitative models, because without quantitative models we'll not be able to handle the data sets. On the other hand, what you just rightly said, that biology becoming math driven, I think this was supposed to happen because if you think about the biological systems in general, right, you see a natural sense of quantitative, quantification and all playing certain role, because the biological systems are also very complex. At the same time, you have lots of molecules inside your cell. You have different proteins. You have again other kind of biomolecules which are interacting, and these kind of interactions are sort of also leading to subsequent interactions, which finally leads to certain outcome in biological cell function. So, and there are a lot of redundancy as well. So you have biological cell, I mean cells have multiple pathways to deal with a certain scenario. So in such situation, if you think about going towards understanding this at a systems level, we need mathematical models. Not just because the data is becoming high throughput, but rather, actually, the biological systems, they are also a very, very complex, right? So we actually talk about non-linearity pretty much whenever we talk about this kind of data sets or even the kind of models. So I feel that mathematics is very, very needed. That we need mathematical models, and in fact, probably the mathematical models are also much simpler as compared to the real biological systems, because when we are actually talking about mathematical models, we are making certain assumptions. Probably there are inherent uncertainty associated with all the things that are happening inside the cell. That's where probabilistic models are so much popular, because now you can actually quantify the uncertainty in your estimates. So I think both of these concepts or aspects are true. Biology becoming more quantitative because of the improved technology, but biology has always been so complex. It's just we never had the means to measure all this complexity in a single experiment. And I feel more and more interaction is now needed between experimental biologist and a computational biologist, and it's more of a continuous conversation, not a single time conversation. It's not just data analysis that is required from the computation side, but rather computational, think of a computational biologist as developing a model which is going to give some prediction. These predictions need to be then experimentally validated. Some hypothesis will be generated. These hypotheses again will be mathematically modeled and probably studied at a systems level. That would actually, so this kind of back and forth, going back and forth, will actually give us answer to a lot of unsolved biological questions. So that's what I feel.
Okay, I got quite a clarity out of it. So if you, I mean, you said somewhere in a few minutes back that you are that for the predictive model that what will happen in future, you are using a neural network, right? So uh, many of our young viewers and many of our student undergraduate student, they
Mhm.
actually eat sleep drink AI/ML continuous, right? That's
Yeah.
what in their mind, right? So I think they will get excited to know that actually you are using a autoencoder type thing, right? So it's a variational autoencoder, right? That's what
Yeah.
I think you said,
Yeah.
right? So those things, right? And many of these students must be using it for other purposes. So as you are a practitioner, so
Mhm.
how AI/ML you think is actually helping you in your area?
I would say that usage of AI/ML is definitely very much needed, and it has been transformative also. And one of the thing is definitely, as I said, there is a deep big data explosion, right? So in biology, you have so much of data being generated every day that these data sets are easily amenable to models like deep learning, because we know deep learning are very data hungry kind of models. But at the same time, I feel there is a lot of caution that one need to actually follow in terms of choosing where to apply deep learning and how, because again we are talking about biological systems. That means there are certain constraints, there are specific type of I would say interactions that we need to pay attention to. So it's not just we can use off the shelf deep learning models or off the shelf AI models directly onto this kind of biological data. So that is where model development also plays a big role. You have all, so for example, I'll give the example of variational autoencoders, right? So people developed it in the field of computer vision back in 2015. What actually happened is that they came up with a generalized framework which was suitable for image datasets, but same kind of model cannot be directly applicable to, let's say, your gene expression data. For that, you need to come up with different types of encoder, different types of decoder. Maybe you need to model, first of all you need to think about your data, what kind of distribution it is following, and then apparently for your suitable distribution, then you need to model your neural network or come up with your neural network model. So I feel that paying attention to biology is very, very important. So it's not that being ignorant about the biology is going to help you in the long run. right? So AI/ML is definitely very important. I think they have found a lot of applicability in analyzing this or solving this biological question. But at the same time, we need to pay attention to biological details. So that means biological principles and constraints should actually inform or guide the model towards better prediction.
So, as you rightly pointed out, that it is not about only crunching numbers,
Yes.
right? At the same time, we have to keep into mind that this what is happening within the domain or constraint of biological principle in which the life
Yes.
works. We
Yes.
may know the principle or we may not know all the principle right now, but there are obviously the physical principle which are controlling life, so it has to be looked into that. So
Yeah.
this brings me something in my mind, that as you said at the beginning, when you were in Jadavpur, you had no clue about that biology is getting mature in this direction, right? And you moved into computational biology, you're doing a PhD. As the problem came to you, got excited.
Mhm.
You must have been very excited at that time, and you are
Yes.
working at that time with the people stalwart at the cutting edge. But at the same time, you must have some difficulties in terms of academic and professional difficulties while you are transitioning from that, right? The transitional problem, right? So
Yes.
can you share some of those, that what may be the difficulty that you personally faced, so that our viewers who are trying to, people who are not from biology background are thinking to move forward into these areas, get prepared.
Yeah, absolutely. I think I will give some personal examples. So, one of the difficulties that I faced, of course, I did not have a formal training in biology. And as we know, like biology, it comes with a lot of terminologies, right? So, these terminologies, one need to be familiar with this terminology. So, initially, when during my PhD, when I started working on genomics data, I actually was bombarded with a lot of new terms that actually are related to biology. So initially, at least for first few months, I had to put myself in a
Struggle.
yeah, I was a little bit struggling with all these new concepts and everything. And but eventually, like, because ultimately I think curiosity is very much important when you are pursuing research in any field. So I was curious that what is going on, right? So again, talking to experimental biologists was also sometimes very helpful. They could explain some of the concepts to me, and reading review papers, that's what also helped me, because there are nicely written reviews about this kind of biological concepts where you get the gist, you get the biological concept. So it's not about the terminologies, but actually the biological concepts that are going to be helpful in the research in this interdisciplinary field. So that, I would say, was one of the challenges initially, because I was not fully familiar with the biological principles all the time. And the second thing was also, I would say, the kind of approach towards a specific research problem. So, in a computational question, just think of any other computational question from any other domain. There is a set of steps. For example, we know that first we need to identify what are the computational bottlenecks, and then we need to identify maybe algorithmic bottlenecks, and then we propose a new solution, we do some benchmarking, and then we are done. But this is not the case when we actually are working on biological problems. We still, again, as a computational biologist, I still have to go through all these steps, that I need to find out the computational or algorithmic bottleneck for solving the problem. I need to validate my approach. I need to come up with the approach. I need to validate my approach and benchmark. But then I think the more important question comes in, that yes, I have a new model, but is it going to tell me something new about the biological system I am working on? Uh, and there are two things to it, or two ways to do it. One of them is, we can first take some very much representative system which have already been thoroughly characterized, and we might ask the question that, can our model still sort of this very well characterized knowledge, can my model recapitulate them? That is the first thing. That is a sanity check, that yes, you are not just crunching numbers, but you are actually able to find biological insights in your systems. And then comes the actual case study, right? You take a biological system, you apply your model, and then you try to sort of look at the results. You bring or make up your own hypothesis. You try to establish your hypothesis, and just like the way research is done in biology, right? So still have to understand whether this is giving us any novel biological insight or not. So this was another thing that I had to learn over time. But I think in the end, it gives us a broader aspect of doing research, that research is not unidirectional, it varies from one field to another. And maybe an amalgamation of all these kind of steps or all these kind of approaches actually leads to better, I would say, better kind of exploration of the research question. So these are the things I think. Both these challenges are, I would say, it's not very major challenge. Even if someone is not coming from a biology background, they should be able to navigate if they are curious enough and if they're hardworking enough.
Thanks, Ami, for spreading out, sharing your experience. And a joke comes to my mind about the terminology. As we say, biology is a subject where division means multiplication. Right? So sometimes somebody coming from outside, it can be
Yeah.
sometimes puzzling what people are talking of, because right.
Yes.
But eventually, as you said, if you are curious, you take it as a challenge and learn and do that. So that this
Absolutely.
brings me to the last question from my side in this discussion today. Is that, see, you are joint faculty in bioscience and bioengineering, at the same time computer science, right? So in you must be meeting undergraduate students from computer science department, right, who want to eventually, who are excited about all these things happening in biology, because we know
Yeah.
biology is a very happening space right now. So they want to eventually move into that in academically, right? And
Yeah.
similarly, I think there are many other people in my campus also. I see people from other engineering background, physics, maths and computing, even from chemistry undergraduate student. They realize that in almost whether talk of academics or in industry, biology and life science has become a major thrust area in human
Yeah.
knowledge creation. So they want to join that, right? They want to move into that. So as you, if I can, you allow me to call you, an outsider moves into the domain of biology,
Yeah.
and you have your experiences to share. So what will be your recommendation for this undergraduate student who may be in the second year or first year, that how they should prepare themselves, what they should read additionally, or something, what they should focus on, so prepare themselves? I think that will be helpful for our viewers.
Yeah, absolutely. I think this is one of the most exciting time to work in biology, right? So with all these innovations that are actually happening, if you see, I think biology is a specific domain which is also sort of like accepting, very much accepting of people from outside. As you rightly called me an outsider, I definitely agree that biology is a discipline which is actually a very good place for outsiders to contribute something meaningful, and that is where I think it's very exciting time to dive into quantitative biology also, not just from the people with background in computation, but also physics, chemistry, as you rightly said. They are actually contributing to biology heavily, and that is where I think, at a very broad level, all these subjects are becoming very, very interdisciplinary. We need multiple people from multiple discipline to work together to solve these bigger biological questions. So, for some undergrad students, again, if they are interested in quantitative biology, I would say they need to be prepared in terms of few different aspects. So, one of the aspect is their mathematical and statistical foundation. So, for example, concepts in linear algebra, probability, these should be like the foundational knowledge for them. So, they need to prepare very well in these subjects. Programming is, I think, bread and butter for any computational person, and I believe some amount of programming is going to be required, I think, for researchers across any discipline, not just in biology, but across any discipline. So definitely a good knowledge of programming and concepts of programming is very, very essential. Of course, now one might argue that with all these AI agents like ChatGPT or anything, so programming is becoming redundant. Like people don't need to learn programming, but I still think the way of thinking about how to write a computer program or this algorithmic thinking is what is very much needed. I mean, of course, there will be innovations in AI, but it can never replace human intelligence. So these are two things I think, as someone in computer science department or physics department or maths department, they will already have these subjects in their curriculum. So they need to study all of these very well. And concepts of machine learning is definitely a plus, or they can also learn as they move into this particular field. And machine learning is again, it's a very broad subject, and every field now is applying machine learning. I think biology is no exception, as I said earlier as well. Now, regarding, let's say, biological concepts as well, I don't think it's a prerequisite to have knowledge in biology to jump into this field. A student, when they move into this field, they can actually learn biology on the way. So they definitely need to learn the biological principles, as I said. The concepts are very, very important. So one needs to be familiar with little bit of concept of molecular biology, cell biology, central dogma of biology, and so on, right? Because these are very important. And another very important thing is the curiosity to know how the data is being generated. Because if we do not understand the underlying principles of data generation, then we'll not be able to come up with very meaningful models. Because the processes that are involved in the data generation, they are also very complex. And once we understand the details of this process, then only we can actually make meaningful contributions in terms of computation as well. So, but I would say that is again part of the research problem itself, right? So if they want to get prepared in a particular direction, or they want to work on a particular research problem in computational biology, they need to learn how the data is being generated, what are the characteristics of the data, and what is the kind of biological system, what are the underlying biological principles. So this can happen as they are actually working on a specific problem. So I would say, yeah, if they have the right mindset that they want to solve this problem, curiosity, there is, I think, no better. I mean, there is nothing better than curiosity. If a student is curious, they can actually solve anything. So I have seen such students I have encountered. I had one MTech student, thoroughly trained in computation, no whatsoever, no relation with biology. But during his MTech, he was publishing papers in NeurIPS. At the same time, he was publishing a paper in nucleic acids research, and he was looking at biological systems himself. He was studying about them, coming up with what are the follow-up experiments he should do after he ran his model. So I have seen people succeed in these cases. So I know that people, if they are curious enough, they can do it.
Okay, yeah, thanks, Hamim, for giving in details about your perspective that how they should prepare themselves. And as you rightly said, and if I remember, when even look into the Nobel laureate list in medical and life sciences, a large
Yes.
number of the people are not trained in biology in that sense,
Yeah.
and outsiders has always played a very important role
Absolutely.
in
Yeah.
learning biology we know now,
Mhm.
and outsider like you are welcome, and more outsiders are needed possibly in this field to
Yeah.
understand life better, right? So
Yes.
thanks a ton for taking our time today, Dr. Hamim, in that and enlightening us and talking about your personal journey from a undergraduate student in Kolkata, then US, and then coming back to India and working in one of the most challenging problem, which still is a major global issue, the cancer biology, how to handle that, understand that. So thank you for sharing your opinion, thank you for taking our time today, and I hope my viewers will be very excited to know more about these algorithms, methods that you people are developing, and they, some of them may eventually get excited to join your league also in future. Thank you for joining
Definitely.
today.
Thank you for inviting me. It was a pleasure talking to you. And hopefully, I have inspired some undergrad students to be excited about this field, which is very interdisciplinary in nature. and hopefully we'll see them succeed in the future. Thank you.
Thank you. Thank you, Hamid.